Can you see the title also? Can you see the title of my? Yes.
No, sir. No, it is not clear. It is cut.
Yeah. So this is personal genomic medicine. So somehow trying to do that. Anyway, other
Yes, sir, visible, sir. You can start.
Yeah. Yeah, friends. Today it's really a very good day. People like me who are interested in understanding the genome and even part of the studies of genomics, where we were not thinking about that is possible in Mysore University, and happy to share that our Mysore University also contributed globally for understanding of the genome and its applications from our department and university. And today, yesterday, the 9th evening, our Prime Minister Modi ji announced that 10,000 Indian genome sequence is completed. That is, Genome India project is completed, and 1,35,000,000 variations are identified, and there are 7 million variations. 7 to 9 million variations are specific to India, which help us to understand the 99 different ethnic groups in India, which in turn help to understand the genomic variations, indigenous genomic variations, and to treat the rare diseases, and many of the drug resistance can be resolved with this kind of genome, our own genome database. So I am happy that we were looking forward from several years, because we were comparing with the reference genome sequence of the databases, but now we have our own indigenous Indian database, and is helpful for that. So that genome database is certainly will going to be helpful for our the title what I'm given, personal genomic medicine. And with this idea, in 2017 only, I proposed a postgraduate diploma in personal genomic medicines for the DBT. Finally, I got through that for the crores of money they are given, and we started in 2000. Sanction in 2018, and we started during earlier to COVID, and we completed 2 batches, supposed to complete, so it will going to be start again, and that has helped us to make our students, train our students to take up many genome analysis. The whole country and even globally is needed the data analyst or genetist or the bioinformatist, the way they can understand the data, interpret the data to make better for the human welfare. With that background, I'll start. Yeah. I'll start this is the outline of my talk. These 2 talk, I'll cover the broadly about genetics and genomics, current medical practices, current practices of genomic medicines, and also we have done some work in our department university, which also help for the genomic medicines, and what are the challenges we have now the genomic medicines, and we'll discuss them also one by one. So friends, you can stop me at any time and ask the questions. So otherwise, this online many times is not good, but anyway, so let's interact. You can stop me any moment and we'll discuss that also. Okay. All of you, can you read this?
Yes, sir.
Why?
Yes, sir.
Sir, why we look the way we look?
What could be the answer?
The
Because
genome.
genes
Genes.
Genes.
and environmental factors and over the like period of time evolution.
Genetics.
Yes, genetics, genes, and genome. And also, I wanted to make it clear in the beginning itself, don't think always that only genetics is going to make everything there. The genes, along with the environment, 50% is the genes, along with the environment, cellular environment and the physical environment. Both are contributing what we are today, what kind of traits we are getting, and what kind of diseases or all kind of things. So let us keep that in mind. No, I'm not going to tell that everything is a gene will going to do, but however, it's a very, very important component in our any organism life. That means genetics is the foundation for all life sciences. Any living organism has a genetic material, let it be RNA in some few viruses or DNA in majority of the organism, and how the DNA regulate DNA is involved in the success of their organism life. Can you see this image?
Yes, sir.
Yes, sir.
Some of you can
Yes,
try to
sir.
do that.
Yes, sir.
So some of you can touch
Visible,
your
sir, it is visible.
yeah, it's visible.
Yes, sir.
Yes, sir.
Yes,
Yeah.
sir, visible, sir.
Yeah, touch your earlobes and it is attached free. Let me know.
It is
You
free.
can think of that also.
It is
Like rolling the tongue and dimple, right-handedness, freckle, clipped chin, even curly hair, allergy. All these are the crossing the left thumb over the right thumb, and all these are characteristic features where the genetic component is mainly involved. Okay, so some of them are we can say dominant characters, like except the attached earlobes, all others are, and except the allergy, all others are the as a dominant features, dominant genes and involved these features. And they are not, none of them are like causing diseases. They are the characteristic feature trait of an individual here. Allergy is a multifactorial. Okay, not only genes, even the environment also contribute for that also. So I have some problem that my title of my slide is not coming there.
Sir, I think you should bring in cursor there. Slide mele tanದ್ರೆ ಬರುತ್ತಾ ಅಂತ.
This one, something is blocking here. Recording.
ಮೇಡಮ್ ಅವತ್ತು ಮೊನ್ನೆ ಇವ್ರಿಗೆ ಹೆಲ್ಪ್ ಮಾಡಿದ್ರು ಸರ್, ಇವರು ಮಂಜುಳಾ ಮೇಡಮ್.
Okay, this is okay now, right? Yes, I did it. I thought if I do that, I may close it also. Yes, done. Yeah, this I can bring it up. Yeah. Yeah, these are the human traits, and you see, we inherit our traits from our grandparents, both maternal and paternal grandparents, and also we share the traits from our parents, father and mother, and also we share the traits from our brothers and sisters. Along this, we do have our own unique traits which has been produced during the gametogenesis of the sperms and egg production from the parents and the fertilizations. So we have all the characteristic features from grandparents, parents, and we share the brother sister, and also we have unique unique characteristic features, and that's why sometimes the people say you are similar to them, and you also have, we also have an identity having our own. So all traits are coded by genes or controlled by genes. And how many types of genes we have? Any idea? Or what are genes? Genes are the one which codes for the protein, right? And
Yes.
some of the genes are not coding for the protein, but however, they are very, very essential to regulate the genes and for the growth and differentiation of the organism. For example, last year, Nobel Prize is given to the microRNA. We have about 6,000 and odd microRNA in human, and they are very, very important. They are just only 22 nucleotides, like tRNA and rRNA. They are the non-coding genes. They are very essential for normal development. If they have some problem, and then it end up with the disease.
RNA non-coding genes.
Hello?
Gunashri ma'am, please mute your mic. Yes, sir. Please proceed, sir. She has muted.
So we generally have coding and non-coding genes. Both are important for normal development, growth and development of any organism, including prokaryotes, bacteria, and the human also. And based on the studies by the scientists, they have been classified based on the structure, function. Genes have been classified like jumping genes. I hope you heard Barbara McClintock, the contributions. Split genes. Jumping genes are nothing but the transposons. Split genes are the eukaryotic genes have exons and introns, which has been very elegantly explained by Philip Sharp, got the Nobel Prize. Edited genes, once the RNA is formed, RNA from the DNA as a template, use DNA as a template, and some nucleotides added to this, the transcript, and they are called edited genes. Overlapping genes, same sequences are being used for two different genes. And polyprotein genes, many genes are involved to making the protein. Nested genes, genes within the genes, means that say gene is about 10 kb, 10,000 nucleotide, and within that in the intronic region, some small genes will be there. They are called nested genes. Repeated genes to overcome the cellular metabolism, cellular activity, for example, for RNA processing and to make a protein, rRNA genes are many RNA genes are needed. For example, in Genopus, where the external fertilizations are there, so many are maternal contributed RNA are there, and they have to become translated after fertilization also. So rRNA genes are more. And processed genes, some genes have been in RNA converted into cDNA, they put back to the genome, they're called processed one. And as I told you, there are several non-coding RNA. They make the differences. They regulate. So there are, to name 12 different types of non-coding genes are there, which are directly or indirectly involved in the cellular processes. Four to 6% of the humanity is suffering from genetic diseases, and there are 4 broadly classified genetic diseases called Mendelian disorders, where single genes are responsible to cause diseases. There you can have autosomal dominant, autosomal recessive, sex dominant, sex chromosome dominant, sex chromosome recessive kind of diseases. And chromosome disorders, I hope all of you are aware of that, trisomy 21, 18, and 13, and they have been discovered associated with the chromosomes in 1960, 1959 and 60s, that has made revolutionized the chromosome cytogenetics. A third category is the multifactorial disorders. Not only genes, even environment also contributing to cause the diseases. Majority of the genetic diseases are belong to these multifactorial diseases. And also we have mitochondrial DNA. The mitochondrial DNA is also involved to make the cellular processes, and if this mitochondrial DNA and the mitochondrial genes are defective, they'll also cause the diseases they call as non-traditional inherited disorder, because mitochondria inheritance is unilateral, or only mother it contributes from the not father, and also it's not undergoing any recombinations. So with this background, the majority of the diseases, as I told you, they are the multifactorial. There are, so for example, here the single gene disorders are cystic fibrosis, and their majority of the genes are responsible a little bit of a sensitive of this environment. Other side, the majority is the environment, and part the susceptible or sensitive genes are involved in HIV, the AIDS, causing AIDS. And we do have multifactorial mental retardations, diabetes, or coronary heart diseases are having 50% of the environment contribution, 50% of the genetic contributions. Okay. Then what is a genome? After knowing that the genes and the characteristic features and how the genetic inheritance takes place, the genome is nothing but entire DNA of an organism, including its gene. Okay, so majority of the organism have genome is the DNA, whereas viruses RNA is the genetic material as the one. What is genomics in that sense? Anybody tell what is genomics?
The branch which studies whole genome and everything, including genes.
Yeah. The genomics, the study of genome,
Yeah.
asking several scientific questions, using some analytical method and using some techniques, and where you can resolve the issue, the kind of questions, whatever you ask, you'll get the complete answer for that. So the field of genomics from 90s onwards, now it has become, we are in the era of genomics now. And 1985, some of the scientists from the Santa Barbara, University of California, Santa Barbara, that is Robert Sinsimer, had an idea that can you sequence the whole genome, and which can identify the culprit gene or the variations which is present in the gene, which can help to treat the human genetic diseases. And like-minded people joined together. Finally, they end up with the Human Genome Project. And 1990, the Human Genome Project has started. Jim Watson was the first director of the Human Genome Project. They planned it for 15 years, three phases. And finally, with the advent of technology, and it has been completed in 13 years. And to start with, the National Institute of Health of USA and the Department of Energy, they joined together. Later, 18 countries have joined together for this, the big science of Human Genome Project. And they revised the objectives of the Human Genome Project. That's identify all the genes, determine the sequence of the complete genome, store these informations in databases, develop the tools for databases, and finally, if there is any drastic changes, drastic kind of situation occurs, you have to address the issues like ethical issues, legal issues, and social issues from the genomic data. So these are the 5 objectives they started, and finally, 2001, Bill Clinton was the American president. He wanted to have, before relinquish his office, to know that how much they have been done, and he asked the public funded genome scientists and the private funded genome scientists. Where Craig Venter started this private funded in 1998, and he told the whole world that in 3 years I'm going to complete the human genome sequencing, and he brought them together and made the working draft of human genome project, and public funded genome scientists have published their data in Nature journal on 15th February 2001, whereas the private funded one, Celera Genomics, published their data 1 day later, and that is 16th February 2001 in Science Journal, and later, after 2 years, they declared that the human genome sequence is fairly completed. That is 14th April 2003. To commemorate the 50 years of celebration of DNA double helical investigation or discovery. So when that double helical structure was been elucidated, most of it teachers are aware. Wait, when it was been double helical structure was first published or discovered?
57, sir.
Yeah, when?
1957.
1953.
53.
53, sir.
Yeah. 1953, the Watson and Crick discovered that, unraveled the double helical structure. To commemorate that in the April 14th, 2003, 50 years of celebration of that. So they announced that we know the genome sequence, complete genome sequence of this. So along with the Human Genome Project and the latest studies of millions of individual genome project, now we have that the data. I'm just going to highlight some of the data from genome sequences. That is, we from between any two of us, we are 99.6% identical. So that means 0.4% we are different each other. Let it be any human being in globally, so any ethnic group also. So 0.4 amounts to the 4 million nucleotide we are different between any two of us here. Those 4 million, minimum 4 million nucleotides are the one making the differences in our phenotype or in our genotype, and that is the one involves some regions like to cause the diseases and so on, to cause diseases and different characteristic features. And Human Genome Project, this is the cocktail project. The 20 individuals have been DNA has been used for this Human Genome Project. It's amounts to 3.2 billion base pairs, the haploid sets, and it's they say 30 to 40,000 genes in the working draft. Finally, now different groups have been simulated that we have 25,000 genes, but now latest one is 21,500 coding genes we have in our genome. This I think I missed one. Okay. The public funded, they did the genome sequencing in a very systematic manner, isolating each chromosomes, fragmenting them, cloning to the artificial chromosome, a small vector, and sequencing them, arrange them. Whereas Celera Genomics use his own shotgun methods of fragmenting the whole genome, whole DNA, and using his own supercomputers, align them and made it overlapping sequences and align them and 888. So the public funded, the individual chromosome they isolated, they have the that is the human chromosomes have been named as 1 to 22 pairs and X and Y based on the size. The longest chromosome is the chromosome 1. It has about 279 million base pairs. It amounts to about 3,000 genes. So that way, they also identified, assigned the genes in every chromosomes. And if you go down the line here, I wanted you to look into that. So the 8th chromosome is smaller than the 7th chromosome, and 9th chromosome again. This is the way. Look at the 11th chromosome. Based on the cytological view, smaller than the 10th chromosome. However, if you look at the size, the genome size is 148 MB compared to the 9th chromosome or the 10th chromosome, and also it almost they double the number of the genes of the 11th chromosome compared to the 8th or 9th or 10th chromosome. So similarly, that if you look at the 17th chromosome is smaller than the 16th chromosome, and the 18th chromosome is smaller than the 17th chromosome, the 19th chromosome is smaller, but 19th chromosome harbors more genes than, even though the size, the genome size is smaller here. So, which indicates that if the genome size is bigger, they need not have more genes. Okay, and also what kind of genes they localize in the particular chromosome also matters. Okay, we know that 21st chromosome is a very culprit gene. 3 copies of the 21st chromosome causes Down syndrome, and it has 303 genes. And later, scientists with the genomic, the advent of the technology and genomic technology, they shortlisted the only 8 critical genes are the one overexpressing the 3 copies, make the mental retardation in the Down syndrome. Now, from the last 2-3 years, people have narrowed down to the 1 or 2 genes and using mouse model, creating transgenic mouse, and they have made it only 2 genes are responsible for they act as a kinase enzyme responsible for causing the heart defects by the Down syndrome. 50% of the Down syndrome kids are having heart defects, and then they they correlated that heart failure, heart defects in the Down syndrome is because of 2 genes or expression of these 2 genes. So that's the latest one on contemporary. And X chromosomes, where the female has 2 X chromosomes and male has only 1 X chromosome, and this X chromosome have around 1,184 genes. The Y chromosome, which make the maleness, and the fertility genes have, and also maleness gene have 231 genes. Other genes are involved in the normal viability and development of an organism. So the human genome sequence data again tells that we have only 1.5, around 1.5% the coding sequences for the protein, and we have about 26% of the introns, which are non-coding sequence intervening within the genes, and about 20% of the long interspersed nucleotide elements, which are now called as a human transposon N1 sequences. They are about 6.5 kb and coding work like the polymerase of the gene. And we have about 300 nucleotide sequences called as sign sequences, short interspersed nucleotide elements. They are about 13%, and many of them are repeated transposons and repeated sequences and miscellaneous and unique sequences we also found in our genome. So this is the overall, they classified the genetic component from our genome. While the human genome projects were almost completing, many contemporary brilliant scientists who were asking questions. If you have the cocktail genome sequence data, it is not going to be helpful for any ethnic group, so it will not going to solve any problem. Then they started many genome projects and many individual genome projects, and just I'm going to list them also because that's another different talks we can make that also there for this. So many projects were launched after the Human Genome Project, but the first genome, the project was International HapMap project. They called as the understanding of genomic variations of the 5 different ethnic groups. 270 individuals belonging to 5 ethnic groups have been sequenced, and variations have been identified in ethnic groups, and they have identified 130 variations which causes major genes there. So common genes, common diseases they identified, and also the individual genome projects like hello, you have any questions there? No? Okay. You can stop me if you have not followed, because all the data I'm not putting here, I'm just explaining each one of them also here. There are 2 individual genome sequences have been completed in 2007. One is the Watson genome sequences completed by Baylor College, and the other company is involved, and they spent about $2 million, and 2 years they spent on that. And Craig went on the same year, he has sequenced his own genome from the Celera Genomics, and he found that his genome size is 2.8 billion base pair when compared to the Human Genome Project, where the 3.2 billion base pair. So indicating that his genome is shorter than average genome sequences, and he identified his the variations in the gene culprit genes, and also he found that some of the cholesterol accumulating genes, the responsible for that are defective, and he started taking 2007 itself the statin to thin the cholesterol in the blood vessels there. So that's a lot of story. Then the other contemporary scientists say that it's not sufficient, so we can, because the technology are also progress at that time. Then they started 1000 Genome Project and ENCODE Genome Project and the Synthetic Genome Project by Craig Venter Group. And then they think that, okay, if you identify this, then we can prove them in the many population. We can identify this kind of variation in many individual population. We call them JIVA studies and genome-wide associated studies. And some of the variations were pertinent to cause the diseases. They think that we can have a personalized genomic medicine that time itself. Then the people have started with the 1 lakh genome project for the Asian 1 lakh genome project, and UK genome project, 1 lakh genome project started. Other progress, like as you know, that genome editing, gut microbiome also major contribution for the human behavior. And later, from last 4 or 5 years, they said that it's not individual country genome sequence, pan-genome sequence is essential to identify the common variant in all the globally human population, and also to identify specific genomic variations of specific ethnic groups. So this post-genomic era contributed every day or every sequencing data contributed. Then they developed the reference genome sequence, that is standard genome sequence. Whoever is doing any genome sequencing, they had to compare that, they identify that which variations are common, which variations are rare, which variations belong to some ethnic groups. So now the major contributions of the differences between individuals and the genomic variations, the changes in the nucleotide. There are 4 different types of genomic variation have been classified. Now we know all the faculty know that the types of chromosome aberrations like I hope you can remember, deletion, duplications, translocations, and inversions are the chromosomal aberrations which causes the involved in many diseases, abnormalities. So within the genome, within the gene where we cannot see under microscope, we cannot see in the microscope where the genomic variations are there, they have been classified as the 4 different types. One is SMP, called single nucleotide polymorphism. One nucleotide is a chain substitute, that is called as SMP. The second category is the SMP. We can distinguish the SMP from the mutation. Mutations are the very rare, and SMPs are the one less than 1%. Any substitution, any changes of the nucleotide is around 1%. They are called as a SMP. The mutation which are very rare, 1 in 1 million, they are called as mutation, rare mutations. So this is the way you distinguish the SMP from the mutations. SMP is also mutation, but their SMPs are very frequent. 1 out of 100 in nucleotide, 1 nucleotide are there. The polymorphic changes we can see there. Next, the genomic variations of microsatellites. I hope most of you are aware of. In 1985, Alex Jeffrey used the repeated DNA sequences. Then it became the DNA fragmenting tool, and DNA profiling the highly repeated DNA sequences. They are the multiples of micro satellites. 3 or 4 or up to 10 nucleotides are repeated in our genome. They are there, so they are unique for particular individuals and the particular family, and they are also genomic contributed genomic variation. Third category is the indels, insertions and or deletions. 100 to 1,000 nucleotides have been inserted in our genome or deleted because of many of the genomic, the mechanisms, the recombinations, the mutations are the one introduced in the genomic variations. So the indels are the third category, the 100 to 1,000 nucleotide inserted and deleted. The fourth category is the 1,000 nucleotide to million nucleotides in some genomes have been added or deleted, and they are called a copy number variation, where the many genes might be missing, many genes might be duplicated. This in large cause the genetic diseases. So there are all these 4 different kinds of genomic variations are responsible to cause many genetic diseases. So they have been not targeted for that. So this is the variation, genomic variations. And now if you identified in individual genome and identified to and correlate with the genotype and the phenotypes, and easily one can treat these genomic variations with available or any new drug. So that is the intention. That is where we are heading towards the genomic medicine.
Sir, in the last slide you were mentioning, what is the 4th one? Like one is SNP, microsatellites, CNV, and what is the 3rd one, sir?
Third one, indels, small indel, INDEL. Indels means IN means insertion, DEL means deletion.
Okay.
100 nucleotide can be inserted and deleted. That category is called indels, small indels, the small insertion or deletions. And 4th category is the CNV, copy number variations. So copy of this, some of the copies can be deleted, some of the copy can be duplicated or inserted. Deleted, insertion, duplication, all the 3 category here. But it's a large piece of DNA comes under this.
Thank you, sir.
Okay, yeah. So genomic variations, these 4 different kinds of genomic variation are introduced every generations at a predictable rate, because the recombination and replication errors are the major mechanism to introduce genomic variation in every individuals, in every generation. These changes are present at conceptions from the beginning, and deleterious and lethal changes are rare. because they're lethal, embryo will not survive, they have been eliminated by the natural selection. And some of the variations will alter the signaling pathway within and between the cells in their physiology or development, so indicating that their abnormality is seen in those individuals. So these variations, genes encoded the proteins are often pleiotropic effect or loss of functions. That wherever the genomic variations are there in that gene, and it can lose the functions. Sometimes if it is a frame shift mutation, and it will the frame of the gene is varying the different kind of amino acids, or it truncated, and the gene will be lost. The gene function or the protein is not going to be formed. It will be lost there, or it might have pleiotropic effects. So these genomic variations are inherited in patterns, and understanding genomic variation, diagnosing the genomic variation in individuals, and treating is very, very essential to treat any kind of diseases. That is the first thing. So understand the genomic variation of the individual, and then the treatment can be done later, or you can prevent those, or you can manage those, the genomic variations. So with this, the globally, many countries have started their own genomic sequences, and now we put all the sequences globally together and develop the reference genome, reference genome sequence called as HG19 or 38. 38 versions of the genomic variation, the standard genome sequences available. Whenever any persons who have a new genome sequences, they have to compare that and analyze that how well it is agreeable there, how are any new variations are there, or the variations of particular ethnic group and so on. So now, as I told you in the beginning, now we have 10,000 human individual the sequences. We can compare in our own Indian databases and global databases how the name variation, whatever the sequence are there, how close they are, how different from there. This is certainly helpful for the management or treat these disease and so on. So the introductions and the preamble I given now. Do you have any questions now? Any doubts you have? Any clarification you want to make so far what I said?
No, sir.
No, sir.
Yeah. So
How?
Yeah, tell me.
Good morning, sir. Sir, how many years genomic variations will take place?
What
I
do you
mean,
mean?
what I am asking is,
Hmm.
there is a general statement.
Hmm.
There are 7 persons are having same character, similar characteristics according to science.
Right.
Is there any same character, completely same characteristics, or a completely a different characteristics according to genomic variations?
Okay, I'll put it a different manner. Yes, I followed your question. Madam, we have only 4 nucleotides. The ATGC, now genome. So they are the language of bacteria or animals or plants or fungi. Any organism, including human, are made up of only 4 nucleotides. They are in a permutation combination of the nucleotides. When this permutation combination of nucleotides, that can have some 7, as I said, out of them 7 individuals might have the same kind of sequences. Okay, I agree with that. Yes, it's possible. Possibilities are permutation combination can make that also possibilities. But to have the same environment, I already told you in the beginning that genetics one side 50% and the environment also 50% to cause any kind of traits and differences or the diseases. So it's very difficult to predict that all the 7 individuals will have the same kind of environment.
Yes, sir.
Okay.
If
So
we compare
indicating that,
Indians, sir, if we compare Indians to Americans or other,
we do have the 99.6% identical, or some individual might be have one gene 100%, but all the genes need not be there. So what I'm telling is that every individual is unique, and you and say, for example, when you compare your own genome with your parents, it's 99.6%. Again, you have your unique sequence, like you and I also have unique sequences.
okay.
So it doesn't mean that if they have identical sequences, they will not have the same kind of environment. For example, environment is also very, very important. For example, you're all sitting in one hall and leave a snake or a cockroach there. What could be your reaction? Right? Immediately, our adrenal hormone is secrete. Adrenal hormone with fractions of millions of seconds, somebody will jump, somebody will run. All those actions will be there. That's the environment. Induced environment, and many gene product communicate to the brain. They take the decision. Some of them, before knowing that, some of them ran away from that region. So environment, what kind of environment we have, that also responsible for action of the gene function also. Okay. Am I explained your questions clearly?
Yes, yes, sir. Yes, sir. Thank you so much.
Sir, I have a doubt, sir.
Yeah, sure. Ask me.
Sir, you are saying the SNP, that is a single nucleotide polymorphism.
Yeah.
How it is helping for analyzing the variation in the genomes?
How?
How it is helping for analyzing the genome variation?
Say, okay. Think that there's one gene have 1 to 100, the number, there's the nucleotide. So this is a common one. And if I sequence your own DNA, that gene, at the 80th position, they change it to different one. Right? Maybe I'll take the example of sickle cell anemia. Remember sickle cell anemia?
Yes, sir. Yes, sir.
Have you heard of sickle cell anemia causes? One nucleotide change in the beta globulin gene changed into one amino acids, that is val into glutamic acid. Okay. Usually our blood cells are oval shape, like this oval shape. Because of one nucleotide change leads to one amino acid change, becomes sickle like this. That is a single nucleotide change. Right? Like that, there are many. We have about 30,000 the 30 million nucleotides we can have globally with all the individuals put together. And also between you and me, theoretically we have 4 billion nucleotide changes. It may more or less also, but globally compared with all the individuals, and we'll have 30 million SMP nucleotide changes also. That's the one, the estimated one here. So some of them are, I'm not telling all the SMPs, all the polymorphism are going to cause the diseases. The people from last 20 years have studied that some of them are neutral, some of them are deleterious, some of them are not affecting it at all. I'll give 1 or 2 examples what we have studied in our own in asthma patients. I'll explain you later also for that. Okay. Understanding this slide is the one. There are thousands of changes are there. How do you quantify them? How do you analyze them? How do you put that? Okay, this is the one then. That analysis, the skilled person, the genetist or the bioinformatics, whatever you call it, or molecular biologist, we have to develop. We have to make them. We have to produce more of such kind of people. That's another reason our MSc genetic course and our diploma courses have started. And most of our genetics, MSc genetics students who are hot cake in the different companies in Bangalore and also MedGenome, nearly 100 students of our own MSc genetics students are doing this kind of genome analysis. So what I'm telling that So we have thousands of variations, millions of variations. How do you quantify? Okay, so look at here. There are now started with one company or two company, and that is 454 companies started. Illumina started beginning. There are hundreds of sequencing company, sequencing technology. There are earlier we was doing. I was doing 1990 when I was a postdoc in Canada. Manually, I was sequencing Sanger method. Now, jealous sequencing is there. They're using different chemistry. There, people have Illumina different, different, and even iron torrent have the only look at the pH. Also, different technology, different chemistry they use that. Okay, so we have AB applied biosystem. It's again different technology they have, but all are giving the same, almost same results. Okay, we have different sequencing tools, sequencing methods, sequencing company. And once you have the data, how do you compile the data? How do you put the data into our system and analyze that? So now we have services like, because our system, our servers may not be sufficient because huge amount of data, terabyte data, and the services like, I hope some of you are aware of this, cloud computing and Amazon Web Services and Genome, because these are the three you can buy for certain time or the space to put our genome for analysis. Okay, so that is for data annotation. We need this kind of the servers or the services there, and we have the sequence, we have the servers. But how do you analyze? We need to have a clinical grade genetist, molecular biologist, bioinformatics, call whatever their life science skilled graduates. So this is the one now, those people are the one analyzing this kind of data there. Even we say that the computer engineers are different and biology students are different, but ultimately they both end together in one. So they make some software, we use the software, we analyze them also. Okay, we are going to the same direction there. So analyzing the one is that diagnosing the genomic variations, very, very important. Unless you know what is causes, how can we treat them, right? Unless you know the problems, how can we proceed with the prevention, the treatment? Our major concern was this, even from last 25 years, in my lab, I asked several kind of questions, several kinds of projects, whatever is limited available, the resources and the funding, we did analyze some of them also. Okay, so just you can visit my own website. Some of the things what we did in limited resources, so I'm happy that I satisfied that we could able to contribute for understand the genomic variations and for further the treatment and prevention and so on. So this is what we need is that we have to generate a skilled persons. The government of India from last 10 years has given a lot of money, skill development for different colleges. I don't know how much it has reached the real person, but this is very, very essential. Not only just having foundation or fundamental, we need to have a skill to analyze them. So this is the genome analysis very important, again, using different kinds of softwares. Okay, let me go back. Now we have, yeah, have any question? Now you can clarify if you have any kind of doubt there. Okay, can I proceed?
Yes, sir.
Yes, sir.
Okay, we have now with the genomic data or biochemical data, we have lot many the markers. How do you say markers? They are repeated. They are seen many individuals, so we consider that as they are highly important markers, like a polymorphism. 10 million polymorphism, the human genome sequencing, even I say 30 million markers, polymorphism are there. Gene expression profile and microarrays for 25,000 gene transcript can be 25,000 genes at one shot. We can understand that also. That kind of technology, transcriptomics available. Proteomics also available to make the specific protein products, and also metabolome or metabolomics, small molecules which are very, very essential for regulations of the genes and the metabolic activity they are needed. So not only the gene or the SMP, even these transcriptomes or the metabolomes are also important to understand the genetic diseases.
Sir, in your earlier slides, the metabolomics, you were mentioning small molecule metabolites. Which kind of molecules it is?
Some amino acids, for example. Yeah.
So how they are specific means they have some specificity. What kind of specificity they show? Like amino acids.
Well, some of the metabolites are like, remember transcriptome factors? To transcribe a gene, at least 10 transcription factors along with the RNA polymerase, even microRNA and these metabolites. All these are needed to refine transcribing the RNA from the DNA, RNA transcriptions they are needed. So, broadly, when I say about 5,000 metabolites are been identified, they are involved in. For example, calcium.
Okay.
Even
So
acidylated. Yeah.
those metabolites which are involved in transcription or translation, they are considered in this. Yeah. Okay.
Yeah. So I'm talking about if you take one cell, eukaryotic cell or prokaryotic cell.
Yes, sir.
What you see in the prokaryotic eukaryotic cells? Genome,
Yes, sir.
RNA, transcriptomes, proteome, and the metabolome. In one cell, these are 4 different components are there, and they are directly, indirectly, many processes we are not aware of, but however, we are strongly aware of that all these 4 are involved in any kind of cellular regulation or cell metabolism for their life, growth and differentiation life.
Got it, sir. Thank you, sir.
Yeah. So, I mean, what I'm telling that with several studies, several data is available, they have said that, okay, these 4 are very, very important. And even if some of the metabolites are not there, and it end up with the disease. If the proteins are not being active, even the protein are there, polypeptides are there, but they are not been active form, and there's a defect. Or transcriptome is there, the transcriptome is not processed and is also affected. And DNA, there's the variations are there, then it's not good. So all these are directly or indirectly or involved in any kind of variation or the disease or the abnormalities of the cell or the individual. So broadly I'm talking about. So they are shift to personal care. We need to know all of them in an individual or in a family. So that way you can make a personal care or the personal medicine for betterment of the individual. So I'm just giving all these other my preamble studies here. Well, we have, if you take up the genomics now here, genetic testing with a single genes or panels by single sequencing. That means, say for example, if doctors identified this is a family have the breast cancer, either the family, the extended family aunt or many people have the breast cancer, then you can go for only breast cancer gene sequencing, identifying which genes are defective. Accordingly, we can think of monoclonal antibody for that particular gene to treat them also. Many success stories are there for breast cancer treatment. Exome sequencing of all the coding DNA, now it's cost about 12,000 rupees. You can go for it with this called next generation sequencing. And genome sequencing of all the coding, non-coding DNA is about around $1,000 or 70 lakh rupees, depending on how many X of the genome sequence is needed. So what I'm telling here, we do have now the panels, we do have the diagnostic kit like single gene identification, small panels of 5 to 10 genes, variations, large panel up to 100, clinical exomes. You have a clinical phenotype. They say that this to produce this clinical, which are the genes are there, they look into the variation of the genes, they make it. And whole exomes, all the coding genes, and whole genome for all the DNA of that. So there are methods are available, pipelines are available, depending on the necessity for the individual or the family, we can go for this. It doesn't mean that all has to go for genome sequencing. Okay, depending on the that means our doctors should be educated that okay, oh, this is for this is for this, and then they should suggest for clinical exome or large gene panel to screen or small panels to screen, not for the whole genome to understand the genetic defect in that particular family and the individual. So this is the knowledge our clinicians, our hospitals should have it, should have. Otherwise, in future, treatment of genetic diseases is not feasible, especially in India. I think they are getting up. Some of the institutions are taking up this. I'll come to the last point when I talk about that. So, what did I say so far? The broadly, whatever I know from 100 years from Mendelian genetics to the genomic era, I just cover this, the broad idea about the gene, genome, and genetics. Let us take about let us put it to get to the other side. What kind of medical practices we have now? What kind of medical practices we have? Trial and error method. Even from our civilization, like they were giving some for cocaine for the toothache, or they say fever, some kind of extra they were giving, non-specific, non-selective for everybody they were giving. Somehow the people have survived in the beginning. It's a trial and error method. Our medical treatment I'm talking about even 100, 200, even 500 years ago, humanity. Even now also, the same drug for everybody, it's being given there. So what we call this as reactive medical care. To some extent, with our the serum test, a blood test, or with the clinical test or radiology test, we diagnose them and select some drug. If the drug not working, so when you go for bacterial and viral infection to the hospital, they give some antibiotic. Doctor asks you to take 3 days or 5 days. If it is not subsidized or if it is not cured, and come will change the next drug. So sometimes it happens that the treatment will be given for some bacteria, but it's not that bacteria is not affected with the drug, and you will go again to that. He will switch to the next drug, and again next drug. So it's a trial and error basis. The treatment even today we are undergoing in our hospitals. So trial and error medicines. So they observe and they treat many times more than 50% they are successful because the clinicians are very clever. If you go there with a kind of fever or cough and other thing, thinking that there might be malaria, there might be tuberculosis, there might be other kind of things. All those then go all the 3 different kinds of drugs they will give. Anyway, the patients will recover. So think that okay, I have cured them. Or sometimes steroid they'll give. So somehow our immune system overcome that. Then the doctor says good. We say that. So success is there. Then say okay, we are done it. There are many failures are there. Then they change the drug from one drug to another drug. Okay, so we are still in the trial and error medicine only. We are we have a drug for many: hypertension, heart failure, antidepressant, cholesterol drug, asthma drug. There are plenty, but all are not successful because every individual genotype is different. Because I already told you 4 million nucleotide are different at. We never know which one is it's affecting here. So effectiveness of drug is not 100%. Effective drug is not complete. About 7% of patients die because of this. And differential drug efficacies are the same symptoms, same findings, same disease, same drug may not work because they do have different resistant gene, different SNPs, different copy number variation. So we fail to distinguish them, and that's why for some people, drug may not work here. So the drug, if you give more drug, it will have more adverse effect. If you give less, it will be ineffective. So therapeutic window, sometimes they will give, see, if the all permutation combination of the concentration of the drug they will give will have many kind of the effect because we do not know drug metabolism in that particular individual, because genotype is also important to understand to understand the drug metabolism in these individuals. We do have many healthcare technologies like medical record, digital medical record. Whenever you go there, so they will enter in the card, they will give the card, and they make. We have a serum biomarkers, genetic differences, gross anatomical features like imaging. They take all kinds of images, even mRNA expressions also they look, and statistical evidences. We do have all these things, but it's not complete because this is with some data, but genetic data, genomic data is lacking there. There has to there. So focus on the clinical needs is bedside to bench and the bedside. Means, so we should know what is the kind of problems are there. Then the problems should be of the clinical problem. It should be analyzed in the bench, the research. Then they have to again go back to that. Okay, this is the kind of defective, and this is the treatment for them also. So many times, with the due respect to the clinician, I'm telling here, they get all the tests done, and they will have the data, and whatever their knowledge they have, that based on that they will give the drug for us, and they cannot understand all the informations. All the data. Maybe sometimes they make block or red. They diagnose the people, and with his own idea, with own experience, he gives that. With information he cannot, but he act and he gives the drugs. So that is the way our present medical care is by trial and error method only. Another 10 minutes. After 10 minutes, we'll after completing this concept, we'll have break there. So now we discuss about broadly about the first aspects about the genes, genome, and the genetics, how it is genomic studies, genome sequences, availability of that. The second one is present the medical treatment, how we are getting the treatment like trial and error method. And also now, if not in India, or if not in all the places, at least in some places, the current practice of genome medicine is already available from last 10 years. The FDA from America has been cleared some of the drug to treat this particular genomic diseases. Many companies are giving diagnosis and the treatment also there. So let us understand what is genomic medicine, personalized genomic medicine. This is my main topic, what I'm supposed to talk here, but with a preamble of whatever the two concepts are discussed here. It's a genomic medicine is the use of information and data from the patient genotype and the phenotype. Genotype means complete sequencing of the data, understanding the genomic variation, which is the culprit gene is defective there. After knowing the clinical phenotype and the genetic genotype, knowing that what the genes are defective, genetic variations, and the clinical phenotypes what the patient has, both have to have the data, and based on that we have to make stratify the disease. Oh, this disease is multifactorial. This disease is involved in a signaling pattern. This disease is the one dominant inheritance. This is because of two defective recessive gene. So one can stratify the disease using this data, and based on that, with available drug, is a gain of function or loss of function. If it is a tumor suppressor gene or suppress, they can give that tumor suppressor product there. So activate them. If the oncogenes have been activated there to suppress the oncogene, the drug can be given there. So that is to stratify the disease, select the medications, provide the therapy, and also initiate the preventive measures for this family or the individual that is suited to that patient at the time of administrations. Genomic medicines, understanding complete clinical and genomic data, so we can prescribe the medicines and also look for this, how it works, and prevention measures also can give to the family. It should not, such kind of the defective person should not be born in there also. That is the genomic medicine. Genomic medicine can also be described as providing right medicine to the right patient at the right dose at the right time. Okay. This genomic medicine make an impact in the field of oncology, pharmacology, oncology here in the cancer, expression of the tumor or suppressor, tumor suppressor gene, or expression, activation of the oncogenes there. And pharmacology, drug resistance will be there, and rare or diseases or infectious diseases. So there are three things. One is drug resistance, another activation of the suppressor gene, inactivation of suppressor gene, tumor suppressor gene, and activation of oncogenes. The major things are there to cause, if you think about the cancer there. So the genomic medicine certainly will be helpful there. Does one size really fit all? That means one drug can be solve all the problem there? No, because everyone is different in their genomic variation or genomic ability there. So genomic discipline that uses genetic information, knowledge of the individual environment, and to customize the patient's health and treatment plan. And again, the individual environment is also important. What kind of lifestyles and the food they take also one has to consider for this treatment there. It's a new comprehensive integrated approach. Genomic medicine is a new comprehensive integrated approach, which this will help to prevent many of the chronic diseases. The genomic medicine or diagnosis help for the early detection of disease risk, and individualized treatments plans are possible with these genomic medicines. And it can also predict the toxicology of new drugs and the risk in the family. Genomic medicine throughout the human life can be given. For example, let us start from the childhood, the behavior of childhood, the genes involved in the artistic or the intellectual disability behavior can be identified. In adolescence, polygenic risk scores can be identified in the germline like cancer, or at the pregnancy level, at the infancy level, all levels of the throughout the human life cycle. Genomic medicine, if you know the defective genes and the variation, it can be given, it can be treated. Genomic medicine cycle starts with patients and the family and description of the phenotype, identify and giving different identical genes, and characterize the mechanism from DNA to phenotype, what genomic variations cause this kind of phenotype, and the genomics in the clinic. All this help for the patient and the family. We are suffering with this kind of genetic diseases to treat them, prevent the diseases in the family. It's a collaborative team practice, not a single doctor or the genetician or the bioinformatics person can do that. 5 key players for the genomic medicine. Patients, one has to diagnose the clinical phenotype of the patients and the genetic phenotype. Principal care provider, the hospitals here. Clinical genome specialist, the doctor should also aware the what is genetics, what are the genomic variation, how the genomic variation can be treated, helpful for this. So now, some of the young doctors are very, very keen. They are aware of this. I'm happy that even we had one project we wrote with the doctor clinicians ourselves, and very, very knowledgeable, because their idea is certainly helpful. Maybe I'm going to tell you one of the example what we have done here. And also laboratory genomic specialists and the payers, the who pay the money for this. All are needed, all need to discuss each other and analyze the data, repeat the analysis, and then treatment can be done. And sometimes this practice is governed by the biological as well as statistical principle. One should be aware of this, otherwise we'll be end up with the wrong treatment there. Personalized medicine linked with observations. You make observation, suggest for trust, different kind of trust, and the actions will be there, and predict. We expect that if you give this, the predictive response, that means the defect, whatever the behavior, whatever the kind of the secretion of the RNA or the changes can be predicted, and that is the one. Breaking the cycle of trial and error medicine is the genomic medicine. Genomic medicine has been 10 years back, has called as era of personalized medicine. Barack Obama approved in the FDA, that is about 8 of the 41 new drugs in 2014, and which have the efficacy, safety, or pharmacokinetics. After that, several drugs have been approved to treat human genetic diseases with the as a genomic medicine manner. There are many tests are there: Oncotype DX, MammaPrint, AlloMap, and tissue of origins like microarray test. We can identify what is the defect, and there's about 550 genes can be studied here. Even AlloMap, about 11 genes can be studied here. This helps in the heart transplantation of the patients, or the breast cancer, about 70 genes can be screened. Which gene has a defective, or which of the genes have a defective, and which pathway these genes are belonging in that, so they can treat them also there. So there's different panels of the gene defective, identified defective genes are available, and these have been used to test and for the genomic medicines. HER2 is for breast cancer. There's a drug, the selection of Herceptin, and many like colorectal cancer or chronic leukemia. There already drugs are available to treat the breast cancer. There's multivariate analysis. Even BRCA genes, they give a monocular antibody for that also. So there are certain well-defined drugs are available for a particular gene or the variation of genes. To whom the genomic medicines? Not for everybody. Those that preferred a proactive approach to healthcare and longevity. Somebody has in the family, they are wanted to check that, then they can check that and they can have a preventive measures. Or if there is, for example, the Angelina mother had the BRCA mutation, her aunt had the BRCA mutation, she has taken, and she also had the BRCA mutation, she has started taking the monoclonal antibody treatment. She is normal now, still she is a model there. So that is the kind of thing. Allied athletes' predictions of athletic performance and sports injuries. So there are, we do have specific genes responsible for fast muscle movement. And if you identify the athletes who are going for the Olympic or the Asiad, if you can test them and we can suggest them also, so whether they can go or not. Concerns for hereditary diseases, inherited diseases based on the family history, prenatal testing. And some of them are exposed to the environmental exposure like Bhopal gas tragedy there, and what kind of gas, how they cause us in the which metabolic pathway. And diet and exercise customization of the individual based on the genome, microbiome, and epigenome. It's also possible. So now the microbiome is also very, so what to eat, that will tell you what you are, so how you are. So genomic variations, microbiome, gut microbiome, and also epigenome, all these are put together, we can make a better treatment and manage the person who really have the genetic defect there. Genomic medicines in the clinical practice, prevention and effectivity is also possible. There are many drugs are available in our system there. And safety. And if the drug is there, it will not have any side effects. It's been proven in the different model systems, and they already